Hello.
Here, how are you?
Good, how are you?
Fine, thank you. y en el 2011 su doctorado también en Toulouse, ya estudiando la interacción entre microbios, bajo la dirección del doctor Bercard. Después hizo su primer postdoctorado en la Universidad de Wisconsin con el doctor Ann, del 2011 al 2014. Un segundo postdoctorado ya más corto, del 2014 al 2015, en el Johns Hopkins Center en el Reino Unido. En el 2015 empezó ya su estancia en el CNRS en Toulouse como investigador asociado, y del 2020 hasta ahora ya es investigador en el CNRS también en Toulouse. Pierre ha participado activamente en muchas revistas, ya sea como revisor o como editor. Actualmente es el Senior Editor de Molecular Plant Microbe Interactions. Es editor invitado de Plant Cell y revisa artículos en muchas revistas internacionales. Algo que me llamó la atención del CV de Pierre es que realmente tiene mucha comunicación de la ciencia, muchas entrevistas en radio, entrevistas en periódicos, en revistas, todo para difundir la ciencia, y es algo que a veces no se ve muy frecuentemente. También ha dirigido postdoctorados a estudiantes de maestría, estudiantes de doctorado, y tiene una gran actividad en docencia, da cursos básicos también en la Universidad de Toulouse. Y de los premios que ha recibido Martes, solamente voy a mencionar dos, los más recientes. Es uno de los cinco ganadores del Rising Star in Plant Science que otorga Molecular Plants, y también ha sido galardonado por la EMBO como Young Investigator en el Young Investigator Program. Evidentemente, Marc ha contribuido importantemente al estudio y al estudio de la evolución de la interacción de las plantas con microorganismos, y ha publicado 52 artículos. Actualmente tiene 3 en revisión que están publicados en bioRxives, y su trabajo ha sido muy relevante y bueno, ha sido publicado en las revistas más reconocidas a nivel mundial, y donde claramente Marc ha dejado clara su contribución al entendimiento a nivel molecular de las interacciones planta-microorganismos, que es precisamente el tema del que nos va a hablar hoy. Marc, thank you very much for accepting the invitation, for being with us yesterday with the class with the students, and today for the seminar.
Well, thank you. Thank you for the introduction. Thank you for the invitation. And although my Spanish is really rusted, I got kind of what you said. It sounds very nice. So thanks, thanks for the introduction. So I'll share my slides now. I mean, we're all used to Zoom, so it should work smoothly. Do you see my slides?
Yeah, that's okay. We can see it.
Great. Okay, I move myself up here. Good. Okay, well, thanks for having me. I'm really excited to tell you a bit about what we are doing in my lab here in Toulouse, France. It's really a pleasure to talk with you because for a number of reasons. First, because I really like interacting with new communities, and I didn't have a chance yet to interact with people at UNAM, and also because and I've seen names in the audience. I have a number of very good friends at UNAM, like Damien or Osvaldo, and it's a great pleasure to share my science, their science that they know well, with all of you. So today I'm going to give you sort of an overview of the work being done in my lab, which is really focusing on symbiosis, plant symbiotic interactions, so between plants and a number of different microorganisms which are in their microbiota. And today I wanted to take a specific spin, or give a specific spin to my talk, looking at transitions between these symbiotic states. How plants, in a way, learned and keep learning to be symbiotic. And that's what we will discuss all together today. I have a number of slides. If I don't reach the last slide, I think it's not a big deal. What I would rather like is to have something interactive. So if you have any questions, any comments, please feel free to interrupt, and I'll be happy to discuss with you all. So let's get started with my usual first slide, which is about plant diversity. So I hope you're all familiar somehow with living organisms. Plants are hopefully, you're convinced, one of the main ones because they are the most abundant in terms of biomass on Earth. And this biomass is divided in a number of plant groups. Some are terrestrial plants. We call them the land plants, and you have them here. They are the vascular plants like angiosperms, flowering plants, or ferns. And you have also a group of non-vascular plants, such as the liverworts. And you may recognize this specific liverwort here, which is called Markantia. It's really a model organism now in plant science. And I will be discussing quite a lot about Markantia later on, so please remember that particular species. There are plants living also in aquatic environments, and they're mostly algae. They can be single-celled or multicellular algae. I won't talk much about algae today, but what you may want to remember is that the most recent common ancestor of all these land plants was actually an aquatic ancestor. So this plant was living on fresh water, very likely. So because plants are extremely diverse and they encounter a huge diversity of microorganisms, they can also form a huge diversity of interactions. And these interactions range from parasitic interactions to mutualistic interactions, where the two partners benefit from the association. In terms of mutualistic symbiosis, we can divide them in two main categories. The ones that we call extracellular symbiosis, and here is an example where you have a fungal hyphae growing around a plant cell. So it stays outside the plant cell. And this is what you see, for instance, in lichens, which are quite common in many ecosystems. And then the second category is formed by intracellular symbiosis, where the fungus or bacteria colonized inside the plant cell. And this is what we call intracellular symbiosis. It's mostly formed with fungi. So you have all sorts of different fungi colonizing living plant cells. But there are also a few bacteria, in particular the one associating to form the root nodule or nitrogen-fixing root nodule symbiosis, which is quite peculiar because in this case it's a bacteria. So this is quite uncommon actually in plants. So huge diversity of plants, huge diversity of mutualistic symbiosis formed in these plants. So if we think about all this diversity, we also have to think about it in a way, in an evolutionary process. Plants probably evolved as non-symbiotic organisms, and at one point learned to become symbiotic. And this step can be in a way called the origin of intracellular symbiosis. Following this first step, we can imagine that other steps occurred to expand the spectrum of symbionts associated with plants. And this is what I call the diversification of these symbiotic associations. You have also a few lineages that actually lost the ability to host intracellular symbionts. So this plant, we call them non-symbiotic. And the most famous one is probably Arabidopsis thaliana, that has completely lost this ability to host intracellular microbes. So through these three different processes, gain, diversification, and losses of symbiotic abilities, we can actually explain the whole diversity of associations we find nowadays on Earth. And basically, my lab has been trying to understand these processes, these three different processes, to explain the molecular basis of these interactions. Can we find the molecular innovations, the gains, the losses that occur together with these transitions, and that explains the diversity of interactions we can spot. So this is the journey we will go through all together now. So before I start talking about the origin of this symbiosis, I wanted to share with you this picture. So this picture is really the picture that hooked me up with symbiosis at the very beginning of my scientific journey. And so in this picture, you see a plant cell here, and a fungal hyphae here. And here, right in the middle of the plant cell, you have the fungus projecting itself inside this plant cell with all these very tiny branched hyphal structures. So this is called the arbuscule, and this is a symbiotic interface. Both the fungus and the plant cell are perfectly alive, and they're actually sharing resources as this interface. So this is, to me, the most intimate and the most amazing structure that you can find in living organisms, where you have actual two different individuals living really interconnected. So this is, I think, the starting point of my interest for intracellular symbiosis. So of course, to reach this stage where you have an intimate association, the partners have to develop this structure. And this goes through three main steps that I will summarize quickly now. So first, the two partners are in the soil, just growing, and they produce chemicals that act as signaling symbiotic signals. So the plant will produce strigolactons. This is a plant hormone, and it activates the fungal metabolism. In exchange, the fungus will produce small molecules derived from chitins, mostly, that induce a symbiotic program on the host side. So together, when they are primed, the two partners start to associate, with the plant forming this infection structure where the fungus can really goes in and colonize the plant intracellular. So the plant is in control of this step. Finally, the fungus will reach specific layers of the roots and develop these highly branched structures I just showed you, which are the symbiotic interface called arbuscules. And in these arbuscules, the partners exchange resources, lipids, carbohydrates from the plant to the fungus in exchange for phosphate, nitrogen, many other type of nutrients and water collected in the soil by the fungus, which are then channeled to the plant cells. So this is mutual reciprocal benefit. So why do we care about this particular association? We care about it because it's just an amazing structure, this arbuscule, but also because 50 years ago, there were two scientists who proposed that actually this symbiotic association probably facilitated the colonization of lands by plants. So originally, I told you the land-plant ancestor was an aquatic organism. I like to see it as an organism growing in one of these lakes. Well, that's actually the sea. This ancestor of land-plant colonized land at one point, and this step was potentially facilitated by the association with the arbuscular mycorrhizal fungi. So that's why I think it makes it quite a fundamental association on Earth. So this is actually well pictured on this drawing from Stefan Rensing in Germany, where it shows the innovations that allowed plants to colonize lands. And among these innovations is the association with fungus. So here it's a mushroom. It's very different from our fungus, but I think it kind of pictures well the concept of terrestrialization facilitated by plants. So this hypothesis was actually proposed in the mid-50s. And the main reasons for this hypothesis were actually three different facts. The first fact is that most land plants, extant land plants, the one living around us nowadays, associate with this concept. So if you collect roots of whatever tree outside your house, you will find this kind of structures in the roots. Second reason is that in most cases, this association is beneficial. It provides nitrogen, phosphate, water to the plant. So there is a benefit. And if there is benefit, you may think that it's positively, or it's under preferring selection. The last reason, and I really love this picture, is because if you look at the most ancient plant fossils we have, and these fossils are coming from the Rhynicher in Scotland, if you make sections of this fossil, you do see that kind of structures that look very similar to the arbuscules I showed you 2 slides ago. So it seems that the association was present in very ancient plants, is still present today, and still benefits the plant. So from these 3 facts, Pierre-Marc and Malloch proposed 50 years ago that this association was probably essential for the first land plants to colonize lands. However, I think I can tell you a different story with the exact same fact. Maybe this fossil is actually something completely different, maybe another fungus or oomycete or pathogenic organism even. Maybe all these symbiosis evolved very recently, independently in these different lineages, and so the benefit is actually something that evolved very recently. So the only way to actually test that this association evolved with the first land plants is to understand the genetics behind and the evolution of these genetic processes. And this is really what we have been trying to do for the last 10 years also. So the first step, if you want to study the origin of any process, is to look at the genes controlling this process and see how they evolve in a given set of species. So here, we built our approach on 20 years of genetics in flowering plants, in angiosperms. Thanks to work in model legumes in particular, we have a set of 20, 30 genes which are essential for symbiosis in flowering plants. And here I just show you a few of these genes. You have the ones in purple that all together form a signaling pathway that we called common symbiotic pathway. And I'll mention this CSP quite a few times. So you may want to remember these genes. They are basically receptor kinases, other intracellular components, a kinase, CKMK, and a transient factor cycles. So all these genes form the CSP, and this is, in angiosperms, a pathway which is essential for symbiosis. Downstream this pathway, there are other genes, like wrinkled, or these 2 alpha-ABC transporters, STAR and STAR2, which are essential for specific traits during symbiosis, like the lipid provisioning of the fungus in angiosperm. So any mutant in any of these 3 genes completely abolish lipid transfer in angiosperms. And we have another 20 genes, which are symbiotic genes in angiosperms. So the first thing we did was to actually take these 20 genes and look at their occurrence in plant genomes and transcriptomes. So we sequenced a bunch of genomes, collaborated with colleagues with additional genomes, and came down to the fact that actually all these genes are very well conserved in all land plants, which means that they were very likely present in their most recent common ancestor that lived on land 500 million years ago. This was the first evidence that this symbiosis may be indeed quite ancient. However, when we just look at phylogenies, that tells us the genes are present, but it doesn't tell us what these genes are actually doing. So maybe these genes were indeed present there, but they were having a completely different function. I don't know, maybe involved in photosynthesis. I mean, I have no idea. So the only way to test that would be to test, like for the angiosperms, the function of these genes in other lineage, and in particular in these non-vascular plants. So to do that, it's easy in a way. We just need to have a good model where we can do genetics in non-vascular plants. And this model, we spent time developing it, but now we have it. It's called Markantia palliaceae. So Markantia, you have a top view here. Markantia palliaceae is a close relative of Markantia polymorpha, which is a non-symbiotic liverwort. So Markantia grows very well on soil, which makes it easy for us to study. If you make a section, it looks like that, with the epidermis here, the lower epidermis and the parenchyma, and these rhizoids. They are like root hair-like structures. The fungus is able to colonize the rhizoids directly and to form this intracellular hyphae, and finally these arbuscules I mentioned and I showed you in roots. So if you look at this association, this section under a confocal microscope, you will see that kind of picture. Well, to get that, you need to practice a lot. I mean, we have a very good engineer doing the microscopy in the institute. So where you see a single rhizoid here, infected with a fungal hyphae, the intracellular hyphae, and finally these beautiful arbuscules. And arbuscules in Markantia are always formed in the same part of the thallus, which is here in the upper part of the parenchyma. That's something also you may want to remember. okay, so we have this species, we can colonize it in a lab setting, we sequence its genomes, we got transformation protocol to work, CRISPR genome editing, all sorts of DNA modules that we can play with. So we have a good model to test the function of our favorite symbiotic genes in a non-vascular plant. First thing we did was actually to look at the transcriptome of this plant in response to the fungus. And here you see a few genes, Markansia genes, which are upregulated in response to inoculation by the fungus, and categorize them in gene ontology categories. You don't really care about that. But what it tells us is that many genes are upregulated in response to colonization in Markansia. And if we cross-reference this data with transcriptomic data obtained in angiosperms, we get a picture like that, where every black square tells you that the genes we found in Markansia to be upregulated during mycorrhizae are also upregulated in different angiosperm species. And you can see here, what you can see here is that many genes are actually upregulated whatever the host plant. Colonization with this fungus always trigger their expression. And we have a few hundred genes that follow this pattern. So we kind of concluded that in response to mycorrhizae, there is a universal transcriptomic response shared across all land plants. If we look more precisely at these gene ontology categories, we do find one in particular which is extremely enriched in these universally upregulated genes. And these genes are involved in the biosynthesis of fatty acids. So all these genes at the end, they just suggest that the plant cell, in association with the fungus, does produce fatty acids. And in addition, we did find genes which are well known from the symbiotic community because they are involved in modifying these fatty acids and delivering them then to the fungus. This is a symbiotic lipid pathway. And these genes also are upregulated in Markansia, like in other angiosperms. So that tells us that plants, whatever the plant, induce the production of lipids, of fatty acids, when it associates with the fungus. And this resonates. This resonates with finding on the fungal side. If we do comparative phylogenomics on the fungal side, comparing genomes of fungi able or not able to engage in symbiotic association, we find that our clade of symbiotic fungi here in the middle, they have all lost the fatty acid synthase complex, this complex that allows production of fatty acids. And this occurred very early on during the evolution of this clade. So on one side, we have plants being able to turn on their fatty acid biosynthesis pathway when the fungus is infecting them. And on the other side, the fungus has lost the ability to produce its own fatty acids. So this is, we think, actually a key for maintenance of the mutualism between the two partners, having one partner completely dependent on the other partner. So of course, this is only based on genomics and comparative transcriptomics, so it's not the kind of absolute proof that this exchange of lipid has been maintained for so long. So to actually prove that, we needed to do a bit of biochemistry and genetics. So first, for the biochemistry, and it was Nicola was a PhD student at the time in the lab, we first look at triacylglycerol accumulation in plants. So this compound is basically a glycerol with 3 different fatty acids fused to it. In the non-colonized plants, you see that we do detect this triacylglycerol. But now when Markansia gets colonized, we see a massive increase in fatty acids. And these fatty acids and this triacylglycerol, they accumulate in the plant during colonization, but they also accumulate in the fungus. So that's what you see here on these photos, where you have this red staining that stains lipid, which is found inside the fungal hyphae. And please remember, this fungus is unable to produce its own fatty acids. So that really suggests that lipids are transferred from the plant to the fungus. We used an enzymatic trick to actually test this direct transfer, modifying the type of fatty acids the plant is producing and then measuring them directly on purified spores. And using that approach, we were able to confirm that indeed the plant delivers lipids directly to the fungus. I won't show you the data here. You can have a look at the paper if you want to understand this process. And we used the trick that was published by others before. We didn't come up with this enzymatic trick. So lipids are indeed transferred from Markansia to the fungus during this symbiotic association. So we thought if there is this transfer of lipids, and if it's really important, turning down this fatty acid biosynthesis during symbiosis should have a massive impact on the association. So how could we do that? We thought we could just focus on the transient factors which are possibly regulating this process and shut them down. And that's the work of Melanie, who is a postdoc in the lab. So she went back to the transcriptomic data and looked for transient factors that were upregulating Markansia during symbiosis, but also across the angiosperm diversity we sampled. And she did find 2 transient factors that were always upregulated during symbiosis. Here you have the promoter GUS of one of them, Markansia wrinkled, which is expressed in the epidermis in non-inoculated conditions. And when the fungus is present, we do have an additional staining here at the base of the thallus where the fungus colonizes, and also in this area, which is the area where we have the arbuscules formed. So it looks like this gene is expressed during symbiosis in arbuscule-containing cells. When Melanie just overexpressed this transient factor in non-colonized plants, and that's what you have here, she did see upregulation of fatty acid genes, like this Cas1 or FAT, which are genes usually turned on during symbiosis. So it really looks like just overexpressing wrinkled is sufficient to mimic this symbiotic fatty acid biosynthesis that we have seen in transcriptomic data. And we also demonstrated that this goes through direct binding of this transient factor, wrinkled, to the promoter of this lipid-related genes. So this is biochemical evidence that there is a link between fatty acid biosynthesis, symbiosis, and this wrinkled gene, but that's not the proof. So the proof, for the proof, we need to do the genetics. And to do that, we used CRISPR. So here on the top picture, you see a section of Markansia, wild-type plants, colonized by the fungus. So you see the fungal structure in green all over the place. So these are opuscules. The upper epidermis is fluorescent because of photosynthesis. So that's just regular staining. Now in a mutant, we get a completely different picture with colonization by the fungus, but absolutely no arbuscules. So messing up with wrinkled and the lipid provisioning to the fungus completely mess up with symbiosis. And from this, we can conclude 2 different things. First, it means because the process is present in angiosperms and in non-vascular plants, it means that this process was already occurring in their most recent common ancestor 450 million years ago. But more importantly, and I think that's really the take-home message here, it means that the symbiosis itself was already present in these first land plants. And this is really the genetic demonstration that this symbiotic association is extremely ancient. We have no idea how this symbiosis looked like. Was probably something similar to what we see today, but I have no idea. We have no idea how these plants actually looked like. But we know for sure that these plants were producing lipids in presence of the fungus to feed this fungus with carbon. And this is quite amazing to think that we don't know the biology, but we kind of know the genetic of these ancient plants. So first conclusion here, we demonstrated now that intracellular symbiosis was an ability gained by the first land plants 450 million years ago. Now that we have this picture very clear, we can start addressing additional questions regarding the evolution of this association. We can try to understand what were the mechanisms that were actually recruited to allow this first symbiosis to evolve. So I won't go through all the details. We have been studying all these genes and many other genes and targeted all the different steps of the symbiosis. Now I just want to focus on one of these steps, which is really the symbiotic signaling involving this cascade that I called the CSP, common symbiotic pathway. So we used a trick here using a reporter that we know to be activated by the pathway. It's a genetically encoded reporter fusing a promoter element to a GUS reporter. When we do that and get medicago roots, so roots of a legume plant colonized by the fungus, so you see the fungus in dark here, we can activate this reporter. So that tells us that in response to symbiosis, this pathway is activated in the roots of flowering plants. We use the same trick in Markantia, and here you see the beginning of an infection occurring, and you see dark blue staining, meaning that this common symbiotic pathway is activated in Markantia in response to colonization by the fungus. We have other ways to show that it's directly Cyclops, which is the transient factor, that activates this element. Then to test whether this pathway is conserved, we did the same than for wrinkled. We generated mutants. I won't show you all the data, but we have mutants for all these 3 genes here, and they all give us the same phenotype, which is like for these different CKMK alleles, a complete lack of mycorrhizae. So in empty vector lines, most of them, or at least 50% of them, of the plant is colonized, while in this CKMK mutant or Cyclops or CMARK mutant, most of the plants are not colonized at all. So it really tells us that, like for angiosperms, this common symbiotic pathway is essential for symbiosis in Markantia. And because it's essential in Markantia and in angiosperms, again, we can link it to these first land plants and really confirm that this is a very ancient and conserved pathway. And we have been studying this pathway quite intensively in terms of genomics as well. So on the next slide, I'll just show you something which is, I will refer to 2 papers we have published a number of years ago now, but I think that's really the foundation for the last part of my talk. So I would like you to understand these different steps. So on this picture, I show you 2 different types of plants. I show you plants like Medicago or Markantia palliaceae, which are able to form symbiosis. And these plants, they do have these common symbiotic pathway genes. So the green means the genes are present. If we look at close relatives of these plants, like Arabidopsis or Markantia polymorpha, which have lost symbiosis, they have lost these genes. So there is a correlation between maintaining the association, intracellular symbiosis, and maintaining these genes. If we look at the entire plant diversity, and we have mined almost 1,000 different plant species, we do see that this conservation is correct until we reach other lineages that have lost the arbuscular mycorrhizal symbiosis, but have evolved other type of intracellular symbiosis. And this is what I show you here with different type of species. They are lineages that have completely lost arbuscular mycorrhizal symbiosis, but they have evolved, for instance, the root nodule symbiosis with bacteria, orchid symbiosis, ericoid symbiosis, or here it's a mucromycotina type of symbiosis, which is specific to this Acrometrium species. So it seems that in plants, there is always conservation of these genes for intracellular symbiosis. We call them the universal intracellular and the symbiosis pathway. And it's really what we think, what the plants use to associate with intracellular symbiosis. Of course, we didn't find 1,000 genes associated with this intracellular symbiosis. We just found a handful of them. And these are all in this common symbiotic pathway. So this suggests that in addition to this role in intracellular symbiosis, they also probably activate downstream programs which are specific to one or another symbiosis, like the wrinkle pathway I described for mycorrhizae. So what we have here is this common symbiotic pathways that probably played a role for symbiosis to first evolve, and then downstream this pathway diversification processes that led to different types of interactions. And my lab is now really focusing on this diversification process. I just show a few slides on actually one of these events of diversification that we have studied most. And this event of diversification is the root nodules, nitrogen-fixing root nodule symbiosis. So compared to mycorrhizae, which is found in all land plants, this association is specific to a few flowering plants, the Cucurbitalis, Rosalis, Phagalis, and Fabalis orders. And during this association, the plant forms this root nodule you are probably all familiar with. And in these nodules, they host intracellular bacteria that fix atmospheric nitrogen delivering this nitrogen to the plant. So this is a huge benefit actually for the host plant, because it gets direct access to the nitrogen which is surrounding it in the environment. So this is an extremely successful mutualistic association. And in exchange, the bacteria gets carbon and other benefits. So this is mutualistic. There have been a long debate about the evolution of this association, and I think the debate is kind of settled now with this scenario, which involves a single gain of this association here at the base of these four orders. And then species in these four orders that do not associate with these bacteria, they probably lost this ability during evolution. And the only evidence we have for that comes from the genomics, where we see 3 genes, 2 that we found in our study, and a third one found by colleagues in a back-to-back paper in 2018. 3 genes that are specifically lost in species that do not associate with this bacteria. So from this genomic evidence, we really think that this association, the root node nitrogen fixing symbiosis, evolved here at the base of these 4 orders before their radiation. So if we want to understand how this species transitioned from a mycorrhizal state to a nitrogen fixing state, we have basically to understand what happened here. And one way to do it is actually to understand what was already occurring here in this first nitrogen fixing species. So to do that, we use a comparative transcriptomic approach. So we gathered many plant genomes and RNA-seq data in presence or absence of the nitrogen fixing bacteria in almost 10 different species. They are the species in black here. So we got this RNA-seq data, we projected them on orthogroups, so comparison across genomes, and we identified genes that were regulated in the different species, but also at the different nodes of the phylogen. And we are able that way to reconstruct these ancestral states. And I'll just discuss 2 of the main findings we had. The first one, I realize I probably don't have the slide here. The first one is that there are genes specific to every single species, which are genes activated by the plant, by every single and specific plant when it associates with this symbiont. But we also found a transcriptomic signature, which is shared by all these plant species when they associate with nitrogen-fixing bacteria. And these genes, we have basically 2,000 genes, we think they are the remnant of a 110 million years old transcriptomic program that was active in the first nodulating species. So if we want to understand what happened here in this first symbiosis, we can just ask a simpler question, which is how this ancestral transcriptome did evolve. How did it build to associate with nitrogen-fixing bacteria? So to address that question, there are multiple ways to do it. The way we decided was to look for a genetic rewiring downstream the common symbiotic pathway, because I told you this pathway is really essential for all intracellular symbiosis. So I'll tell you the strategy in one slide. We think that in species associating with avuscular mycorrhizal symbiont, activation of the pathway lead to activation of a gene regulatory network, essential for this association. And during evolution, at the origin of this root nodule symbiosis, this pathway then got access to a different gene set, activating a different gene regulatory network. And this is a network we do see now in these ancestral nodules. So understanding how nodulation evolves is maybe as simple as understanding or identifying what are the genes which are now regulated by the common symbiotic pathway in this species. This is our kind of target. So to do that, it's not that simple, because we will need a way to specifically find these genes and not other genes triggered by other processes. So we use a genetic approach. We overexpressed part of the pathway, either kinase here, other transcription factor, and we made autoactive versions of these genes. So they are constitutively active. If we overexpress these constitutively active genes, we should actually activate this pathway here, and we can measure that by our RNA sequencing. So this is what we have done first in Medicago, which is a species able to form the arbuscular mycorrhizal symbiosis and the root nodule symbiosis. So when we do that, we do find many genes upregulated, and half of the genes we find upregulated in response to the transcription factor. We find them also upregulated in response to the autoactive kinase, which is known to phosphorylate the transcription factor. So this is the proof that actually this strategy does work to capture genes associated with the pathway. And if we cross-reference that with RNA-seq data in response to modulation or to mycorrhizae, we do see that we have quite a nice overlap between the genes we activate with this genetic trick and the genes we activate in response to the symbiote. So that's what we did in Medicago. But Medicago gives us access just to one state, the state where a species is able to form the two symbioses. If we really want to understand what happened during evolution, we need to get this picture, but for many other species. And what Tatiana did, she's a postdoc who has been leading this project for the last 6 years now. She actually overexpressed these two different gain-of-function constructs in 13 different species, covering all the type of symbiotic states we can think of. So first, she did it in species able to form mycorrhizae, and we thought, well, that gives us the gene set linked to mycorrhizae. And from this gene set, we can cross-reference with the genes expressed in species that form nodulation. And doing so, we get access to the nodulation-specific genes. There is a factor we have to correct for, which is that some species, all the species that form nodulation are monophyletic. So to correct for this factor, we'll also overexpress these two constructs in species that have lost nodulation. And by removing the genes still activated in these species, well, we get the genes that are supposed to be specific to the nitrogen-fixing symbiosis. So we did this, ran RNA-seq on all these different species. That was a hell of an effort. And we got gene lists. So I won't go through all the gene lists. That would be really a very, very long seminar, and that's already quite a long seminar. So I'll just show you 2 slides. The first one are the genes which are expressed in species that form mycorrhizae. And we do find genes that people, if there is anyone in the audience who is familiar with mycorrhizae, this gene will sound quite familiar because they are very typical symbiosis genes like RAM2 I mentioned or STAR, which are involved in the lipid metabolism. And then we did this cross-referencing depending on the symbiotic states. And we ended up with this list of genes we care about. And here I just show you a few of them. We did find many transcription factors. We did find signaling genes, which are also well known for nodulation, like this too much love protein here, which is important for the autoregulation of nodulation. And we also found genes important for the actual infection by the bacteria. So we were quite happy to actually find genes we know very well. And we also found a list of pretty much 80 genes that we don't know about. And these genes are now our top target to explain how nodulation evolves. So I don't have the gene names here because there is a company involved and they really didn't want me to put gene names on this slide, but we are now trying to understand what these genes are doing. I'll tell you a little bit more now about some of the transcription factors that we find here. So I guess BHLH-A is not a name you can kind of spot, so I'm allowed to talk about BHLH-A. So there are 2 main questions here. What are these genes doing? And how did they get recruited downstream this common symbiotic pathway during evolution? So I just show you 2 slides about that. First, how did they get recruited? And here I show you one example. So here you have the study of the promoter of one of these genes. It's called NIN. It's a very famous gene in the symbiotic community. And if you compare the promoter of this NIN gene across species, looking at species that nodulate, the one in blue, and the ones that do not nodulate, the one in black, you see that there is this motif in the middle, which is highly conserved in species that nodulate. But this motif seems to be degenerated in species that have lost nodulation. So this is a sign that there is a relaxed selection on this element and that it's probably associated with a trait of interest. And actually in species that nodulate but have duplicated this gene, like in Phaseolus here, you see that one of the copy is actually quite mutated, showing again that there is probably purifying selection acting on this element. And actually this element is kind of makes sense. It does make sense because it's what we call the Cyclops responsive element. That's the promoter GUS we used in Martes and Medicago to show activation of the pathway, because this promoter is targeted by Cyclops. And the fact that we do find this element in the NIN gene exclusively in nodulating species indicates that this connection between the common symbiotic pathway and NIN probably evolved very early on during the evolution of this pathway and was one of the drivers for the evolution of nodulation. And you can find the details about the genetic dissection of this element in this paper, which is still only on bioRxiv, I hope soon online. Second question, what are these novel transient factors doing? And I'll just show you one slide here, where we are collaborating on this functional validation with colleague in Aarhus in Denmark. And this is the work of a postdoc there, Manuel. So Manuel generated single mutants for 2 of these candidate genes to BHLH. So here, and he counted nodules in this mutant and on the wild type. And what you can very easily see here is that the wild type plant form nodules, an average 4 nodules per plant, while in the mutant there is a strong reduction in the nodule number, showing that this gene is important, or these 2 genes are important for nodulation to work. We have single cell data for nodulating plants, and we do find these genes expressed in infected cells. So this is what this weird figure tells you. So these genes are important for nodulation, and they are expressed during infection. So we think there is a link between these 2 processes, and Manuel quantified the number of infection events on the roots of these mutants compared to the wild type. And you can see that there are many, many more infection events on the wild type than on any of the 2 mutants, showing that 2 of the genes we found downstream the common symbiotic pathway, specifically in nodulating species, they are important for infection. And basically, it also validates our approach that 100% of the candidates we have tested so far, 2 genes, are actually playing a role during the symbiosis. So that shows that during the evolution of the root nodule symbiosis, genes got recruited by the common symbiotic pathway to diversify the symbiotic options of plants. And this is something we are trying to finalize now. And that will be my last slide, actually. We are trying to get a good, sorry, my phone is ringing. Okay, my PhD student. So what we have been trying to do so far is to understand how evolution works. This is evolution of symbiosis works. That's really what we have been trying to do very, very hard. But what we would love to do, and that's what we're trying now, is to actually transition from learning, dissecting these symbiotic processes into building novel type of associations, and in particular this nitrogen fixing symbiosis. And this is something we are doing in the context of the ENSA project, which is funded in part by the Bill and Melinda Gates Foundation. And this is really something we are trying to achieve now. And I hope that in a few years, I will be able to come back here virtually with you and to tell you about this nitrogen fixing nodules we have on corn. That would be quite amazing. But that's for a talk in the long-term future. So now I would like just to finish with a few acknowledgements. So here are the main people who have done the work I've presented. This is our group picture. We were in our lab retreat in the Pyrenees near Toulouse. So please come by and visit us. Toulouse is a wonderful place, and the Pyrenees are just nearby, and it's just an amazing site. I would like to thank also our collaborators on these different projects, Philippe and Delphine for the comparative transcriptomic I presented on nodulating species, Martin Parniske's lab for the work on the sick responsive element in the immune promoter, and Manuel for data on the BHLH. Funding agencies, this work was mostly funded by the ANR, the ERC, and the ENSA project. and you all for your attention. I hope it wasn't too long. I didn't really check the timing. I'm really happy to take any questions you may have. Thank you.
Thank you for your presentation. It was perfectly on time. So questions, please raise your hand and open the microphone. Go ahead, Aldo.
Yes. Do you hear me?
Yes.
Yes. Thanks for the talk. So yesterday I read that some of these fungus were able to colonize the nodules formed by rhizobium in some legume plants. So I also saw that whenever this happens, the nodules were not fixating nitrogen anymore. And my question is whether this loss is needed for the colonization or the presence of the fungus is inhibiting the nitrogen fixation.
That's a tough question to start with. So actually, the colonization of nodules has been described in a very few cases. So to my knowledge, this is quite unusual phenomenon, I think. But I think Georgina may have a comment about that. So I don't know what's known about this process, but to me, it's very rarely occurring. So I don't think that it's actually a driver for reducing nitrogen fixation efficiency. It may occur, but my guess is that it's very rare.
Okay, thanks.
Hola.
Hola. So I can tell Gustavo?
Go ahead, please.
Yeah. So thank you, Pierre-Marc, for this great talk. It's quite amazing to see you there. And I have a question about Markantia. So now we give you a big list of new genes you cannot share. Do you think you can rewire Markantia to nodulate or something like this, you know?
Well, to nodulate, I don't think we can. First, we have to evolve roots and then potentially get them to nodulate. One thing we are trying as a proof of concept would be to get rhizobia in the rhizoids. So can we manage intracellular infection by a bacteria in a plant outside these four orders of the papacural clade? So that's what we are trying to do. What we have already managed is to rewire signaling. So we can add steps, activations, and so on in the pathway. This is already an achievement, but yeah, our first goal would be to get rhizobia inside the cell. That would be already quite something, I think.
Yeah, so I have another question. Sorry, Gina. We are really interested in LBD, the lobe domain binding transcription factor. So what you can say about these genes? Are you working on this family? What is the importance for you of this transcription factor in the nodulation or mycorrhization?
Okay, so we do find them. Well, I may have added one of the LBD in our list. I can't remember whether it was on my slide or not, but we do have a LBD in our list that we are studying now. What I can tell you is that it's not one of the LBD 16s that were published by Makoto and Giles in Lotus and Mid-Cargo, and they showed that they are directly done, or not directly, they are downstream NEAN and play a role in nodulation. I mean, a very significant role actually in nodulation. We do find another one in the family, and we think they are somehow all doing the same thing. So we are just now trying to validate that functionally in the nodulation context. For mycorrhizae, we don't have evidence that they would be involved. So they don't show up in the transcriptomic data. In our Cyclops by CMACAMK work, they are really specific to nitrogen fixing species. So we think this is a specific addition, an add-on to make nodules, so not for mycorrhizae. That said, if you start studying these genes in a legume, legumes are weirdly wired, right? So they integrate the two associations. So touching one may impact indirectly the other. So I would suspect that maybe touching LBDs in Medicago or fatherless may actually impact mycorrhizae. But I think that's probably like a legume or nodulating species specificity rather than something really common across mycorrhizal species. It doesn't mean it's not interesting. It means more specific.
So the LBD you talk about, I don't know if you can say so much thing.
LBD4.
LBD4? Oh, okay. So it's a class 2 or class 1 LBD, you know, because these LBDs associated with nitrogen assimilation and not the root development stuff. What kind of class is the? Okay, maybe it's too much.
Yeah, I couldn't tell you. I mean, I just don't remember. We have so many genes that I don't know the details.
Yeah. Okay, so thank you so much for your answer.
May I ask, Gustavo?
Yes, go ahead, Gina, please.
Hello, Pierre-Marc. Congratulations for this excellent seminar. I heard something about this recently in Naples. And one thing that I can tell about Naples that Gustavo did not mention was that Pierre-Marc received a prize there as a young scientist, the Adam Condorossi European Prize. And congratulations for that also.
Thank you.
My question is, well, obviously you demonstrated excellently that the importance of the exchange of lipids between the mycorrhizal symbiosis. But also you have heard, as well as I and many colleagues, for several years, the importance of mycorrhiza to provide phosphorus to the plant, especially in environments that have low phosphorus. And this is important for crops, etc. I really don't know if how the concentration of phosphate was in early times in evolution. I don't know, but can you mention something about this? What do you think about this function or this necessity or this how the fungus can provide phosphorus? Was this important in evolution or not? Or
this is a very interesting question for a number of reasons. So first answer, phosphate was very limiting in the ancestral soil. It wasn't a soil. I mean, you really have to keep in mind that there was basically bacteria growing there, like a bacterial mat, and growing on rocks. So phosphate was extremely limiting in this environment. So that's why we think actually that it was possibly one of the drivers for this association to evolve and why it facilitated colonization of plants. So if we look at the transcriptomic data, we do find phosphate transporters upregulated in Markansia. And there is one report showing actual transfer of phosphate from the fungus to Markansia using radioactively labeled phosphate. So there is transfer of lipid. We have phosphate transporters upregulated in Markansia. So that really points to the conservation of this process. But, and that's where it gets really interesting, the class of phosphate transporter, which is upregulated in Markansia, is different from the one in angiosperms. So they're all in this PHT1 family, but the one in angiosperm belongs to what we call the PT4 family, while the one in Markansia belongs to a clade that evolved specifically in liverworts. So that tells us that the mechanism, the process is there, but the mechanism is different. So maybe phosphate is something that either evolved independently in the two lineages, or that diversified, so evolved once and then diversified following gene duplications in these two different groups. And that's particularly intriguing because if we look at the ammonium transporter, so we know mycorrhizae also helps or facilitates ammonium and nitrate uptake. If we look at the ammonium transporter, which is from the AMT2 family, this one is actually extremely conserved, and it is the same which is upregulated during mycorrhizae in Medicago, in rice, or in Markansia. And that suggests that maybe the main original driver for mycorrhizae to be conserved was rather the transfer of nitrogen than the transfer of phosphate, which is counterintuitive in a way. But I find it quite interesting. I like, when the facts don't match with the original hypothesis. I think that's where science gets exciting. So I think, yeah, probably both processes were important, but it's not only phosphate. Nitrogen was very likely very important as well.
Thanks, very interesting. Thank you.
So people in YouTube can write down your questions and we can read it here. So I will ask a quick question to give time, so if somebody is interested to asking questions from YouTube. Pierre, I have a question regarding the immune response of the plants, specifically in the ones that you overexpressed, the kinase and the transcription factor. There was a downregulation of genes involved in response against pathogens in those plants?
We do not see that. That's something we looked at because that was part of the potential expectations, but we do not see that. And in the upregulated genes, we don't find immunity-related genes, but we don't find them in the downregulated genes. But when we compare the transcriptomes of the nodulating species, we do find a strong reduction of the defense response in the nodules, except in one case when we play with rhizobia that do not, we play with modified rhizobia, and the ones that don't persist, we still see activation of the defense response. So the ones that don't fix, they are killed by the plant, and there at the transcriptomic level, we see activation of the plant defense.
Okay, thank you very much. So another question? Go ahead, Natalia.
Hey, thanks for your talk. And I have two questions. Maybe I should ask yesterday that questions, but I don't know why I don't ask. But the first question is that because polyploidy is common in plants and have a significant impact on evolution, I guess, are there traits that are particularly affected by these genome duplications or this interaction in plants and fungus and other microorganisms?
So that's a very broad question, right? So I'm sure we can find specific cases where there are links and connections. To my knowledge, there is nothing clear when it comes to whole genome duplication, polyploidization, and symbiotic association. So there was an hypothesis at the time suggesting that maybe genome duplication in papillonoids facilitated the gain of nodulation. I don't know if that's an hypothesis you have heard about, but actually, now that we know nitrogen fixing symbiosis evolved much earlier, there is no correlation anymore. So I don't think there is an influence of these processes on genome-wide expansions on symbiotic behaviors. I think the only thing that we witnessed is that our favorite symbiotic genes, the one from this common symbiotic pathway, they are almost never duplicated in genomes. It's very rare. There are a few exceptions, but they are almost never duplicated. Although these lineages have experienced massive whole genome duplications, tandem duplications, and so on. So I think these genes are under strict dosage control. They have to be limited in the genome, otherwise it leads to defects. Why, I have no idea. But this is quite peculiar. They belong to gene families which are not expanded, which is just weird for plants. Well, that's the only thing I can say regarding these two processes. I'm not sure it answers your question, but that's the only link I can make.
Thank you. The other question I have is how can we add the effect that the change in climate or these other mutualistic or conflict interactions with plants and other animals, maybe like insects, that can affect these, I don't know, these interactions and these other particular traits that may evolve in plants?
Well, you come up with difficult questions. Well, climate change and symbiosis, difficult to predict. We know the symbioses are quite sensitive to environmental perturbations. Too much water, no symbiosis. Not enough water, no symbiosis. Perturb microbial communities, it affects the symbiosis as well. So anything that would impact massively the environment would very likely impact symbiosis. This symbiosis the way we know it now. This symbiosis is 400, the arbuscular mycorrhizal symbiosis is 450 million years old. So I'm sure plants will evolve new ways in the fungus to adjust with the climate. But what the biodiversity of these symbionts we know now will probably get extremely perturbed in what we are experiencing right now, which is global change. For nodulation, it's interesting because there was one link made by René Gertz in the Netherlands, and between the loss of nodulation and global climatic events. So there is, it seems that there is, at the KT boundary, there is a massive loss of nodulation, showing that massive impacts on the geochemical cycles impact nodulation quite dramatically, because we are talking about hundreds, if not thousands of lineages losing nodulation. So that's just an hypothesis, but maybe that tell us that these associations are quite sensitive. They are quite sensitive. So as, let's say, a biologist, I would say climate change will mess up with all biodiversity, so it will probably mess up with symbiosis. And I hope that mycorrhizae and nodulation won't be too affected.
Thank you, Pierre. So I think we will leave it here. So thank you very much again for your participation in the program. It was very illustrating. So hopefully we'll see you in the future once more.
Would be my pleasure. Thanks again for the hi Jesus. Would be my pleasure to come back virtually and discuss more. And thanks again for the invitation.
Thank you, Pierre. Gracias a todos por estar acá. Hasta luego.
Hasta luego. Thank you, Pierre.
Bye.
Hasta luego.